Hello everyone, welcome back to our class. Before we start, a question. Do you think the human body is electrically charged? Some students say yes. Some say no, and others say sometimes yes, sometimes no. In fact, the human body is electrically charged. This story begins with a Dutch microscopist. Around 1660, a Dutch scientist named Jan Swammerdam dissected a frog's leg and carefully separated the muscle connected to the nerve and placed it in a homemade apparatus. He then stimulated the frog's sciatic nerve with an electric current, and to his surprise, the isolated muscle contracted. Although he did not propose that nerve conduction is an electrical signal, this experiment laid the groundwork for later research, demonstrating the existence of bioelectricity. Scientists call the electrical phenomena associated with cellular activity cellular bioelectricity. Clinical diagnostic tools such as ECG, EEG, and EMG record the bioelectrical activities of the heart, cerebral cortex, and muscles at the body surface, which are the summed results of cellular bioelectric activity. The bioelectricity of a single cell occurs across the cell membrane, thus called transmembrane potential or membrane potential. A cell's transmembrane potential includes the resting potential and the action potential. The resting potential forms the basis for action potential generation. Resting potential refers to the potential difference across the membrane when the cell is at rest, if the extracellular surface is defined as zero potential, then the intracellular potential is negative. Using intracellular recording, the resting potential is expressed as the negative intracellular potential. Resting potentials vary among cell types, typically ranging from -100 to -10 MV. For example, mammalian skeletal muscle cells have about -90 MV, nerve fibers about -90 to -70 MV, and red blood cells about -10 MV. The basic cause of the resting potential is ionic diffusion across the membrane. There are two conditions for ionic transmembrane diffusion. First, unequal ion distribution between inside and outside the cell. High K+ concentration inside and high Na+ concentration outside. Second, at rest the membrane is more permeable to K+ and less permeable to Na+ and Cl-, and nearly impermeable to organic anions. Under these conditions, intracellular K+ diffuses out down its concentration gradient. K+ carries a positive charge, and its outward flow increases positive charge outside the membrane, increasing relative negative charge inside, creating an electric field with inside negative and outside positive. The outward flow of K+ cannot continue indefinitely, because the inside negative, outside positive electric field grows stronger, eventually opposing further K+ efflux. When the concentration-driven force and the electrical force balance, there is no net transmembrane movement of K+. At this point, the potential difference caused by K+ efflux stabilizes, and this potential is called the K+ electrochemical equilibrium potential, or K equilibrium potential. In addition, Na+, Cl, and other ions also contribute to the resting potential to some degree. In summary, the resting potential is the stable potential difference across the cell membrane at rest, mainly determined by the electrochemical equilibrium potential formed by K+ efflux. Based on the mechanism analysis, the main factors affecting the resting potential are: 1. Extracellular K+ concentration. At rest, the membrane is relatively permeable to K+, and changing extracellular K+ alters the K+ equilibrium and resting potentials. When extracellular K+ rises, e.g., hyperkalemia, the K+ equilibrium potential decreases and the resting potential correspondingly decreases. 2. Relative membrane permeability to K+ and Na+. If membrane permeability to K+ increases, the resting potential becomes more negative. Conversely, if Na+ permeability increases, the resting potential decreases. 3. Activity level of ion pumps. When pump activity increases, its electrogenic effect strengthens, causing the membrane to hyperpolarize to some extent. Conversely, when the Na plus pump is inhibited, the resting potential may decrease.